Alright, we have a third video because I accidentally hit end on the other recording early due to a cat. I apologize. So, continuing where we left off, we talked about junctions, gap junctions specifically, which led into intercellular communication. How do cells coordinate the activities needed for homeostasis? Directly, this typically is because of those gap junctions. Chemical messengers include things like CAMP and calcium. Calcium is a chemical messenger. I know we don't think of it as such, but it is. Or it can be indirectly in the aspect of a chemical messenger is released by one cell and it will act on the specific receptors of another cell. And that cell can either be close by or it can be in a completely different section of the human body. For instance, insulin released by your pancreatic beta cells in the islet of Langerhans will be released into the bloodstream. It will go throughout the body and it will bind to insulin receptors on cells throughout the body. And when it binds to these receptors, it will facilitate glucose uptake. Basically, this is the mechanism that when we talk about a patient being insulin resistant is at play. This insulin is being released, so they're still making insulin and it's still being released in response to the blood sugar going up. However, when it binds to the receptors, instead of it responding as normal, so the cell taking up glucose, the cell ignores the signal. It's just like, oh look, you're back. Cool. Did you want something? Did we need to do something? What's going on? Instead of, hey, cool, you're here? Let me open up. Let me bring in the glucose. So that's insulin resistance, basically. Now, the why for the insulin resistance, so why do the cells stop responding? That's something else. And how do we get around that? And why do certain things get around that? For instance, if you have insulin resistance or diabetes, they are not necessarily the same thing. Insulin resistance tends to be a precursor to diabetes, but not necessarily a diabetic patient. So if I have insulin resistance, one of the things they'll tell me to do, they strongly recommend, is exercise. Because what they've noticed is patients who have insulin resistance, if they exercise, it's not necessarily to lose weight, but what it does is their body responds to the insulin. So it's ignoring it, bam, it starts responding. It starts, oh, hey, you bound to me? Okay, I'll let it glucose in. Why? What else is going on when you exercise that makes the cells sit up and pay attention? Welcome to research. Okay. Protein synthesis. I've mentioned it in passing. Let's get specific. So, first and foremost, I need some DNA. In order for protein synthesis to occur, I need some DNA. And that DNA is going to contain my code. It will be read or transcribed into messenger RNA. That messenger RNA will move out of the nucleus into the cytoplasm, and then it will go and find some ribosomes where it will be read to make proteins. Now, does it have to go into the cytoplasm? No. It can also go into the rough ER, but it's the same concept. It will move out of the nucleus and then find a ribosome to create a protein. How does our DNA store genetic code? A gene, first and foremost, back to terminology, a gene is a portion of your DNA that is holding a specific code. So when we talk about various genes, we're not talking about the entire complex, we're talking about a specific portion of the complex. And then within that DNA, it will be coded in triplet forms. Triplet form, otherwise known as a codon, is a sequence of 3 nucleic acids. And when those 3 nucleic acids are read, they form a specific amino acid. It looks like this. Here's our gene, a specific section of the DNA. It gets read, and when it's read, all the DNA is grouped, or nucleotides are in groups of 3. These form codons, and this allows us to make the codes for 20 different amino acids. Now, this three triplet coding, because we have 4 different bases, allows for the formation of 64 different codons, or 64 different triplets. Each one will code for a specific amino acid, or a start or a stop. So basically junk code, doesn't code for anything, doesn't make anything. However, we can have more than one triplet code for the same type of protein. For instance, CCC is proline, but also CCG. AUG is a methionine coding, and that is our start codon. If it doesn't code for anything, so if it doesn't code for a amino acid, that's going to tell the ribosome to stop. We're done making the protein chain. You may be familiar with a table similar to this already. This is how we figure out what triplet makes for what protein. For instance, I would start on this side. My first letter is a U. Okay, now I need to find the second letter, so I'm going to go across to the C, because it's UCG. And then I'm going to come down to the G. So UCG. That's going to give me serine. Or if I have a UAG, so UAG, that's going to say stop. Are you going to need to memorize this, be comfortable with this for my exam in physiology? No. However, if you took Gen Bio here, you have done this before. Transcription. Transcription is the process by which RNA is synthesized from DNA. Basically, we can transcribe 3 different types: messenger, ribosomal, and transfer. And how do I know which type of RNA is which type? Well, you look at the little letter in front of the RNA. If it's an M, it's messenger. If it's R, it's ribosomal. If it's T, it's transfer. And that's about as much detail as we're going to go into, except for the fact that messenger is going to carry information from the DNA to a ribosome. Ribosomal is going to help read that RNA, and then transfer is actually going to be what the amino acids are attached to that brings the amino acids to the messenger RNA and to the ribosome. So we need all 3 of these working together in order to actually form the protein chain. To get this RNA, we are going to need RNA polymerase. There are a slew of different RNA polymerases. Each one does a slightly different thing and are involved in our messenger RNA versus ribosomal versus transfer, so we're going to speak about them generically. But this is going to be an enzyme. Remember from med term, anytime you see A's, A-S-E, we are talking about an enzyme. So this is an enzyme that is going to be involved with RNA. The RNA polymerase we're going to mention is the one that is going to bind to what's referred to as a promoter sequence. Basically, this is a section of the DNA that the RNA polymerase binds to, allowing for and triggering the uncoiling of the DNA, which will then allow the DNA to be read. It's also going to catalyze the bonds of the various nucleotides for the messenger RNA. So it's going to allow those nucleotides to chain up together, reading the DNA. So if we have an error with these enzymes, either we don't have them, or we don't have enough of them, or they're not coding correctly, or they got coded incorrectly, so they're not in the right formation, we're going to have an issue with creating protein chains. The DNA will not be able to be read, and the RNA will not be able to link together, reading said DNA. Okay. So when we read DNA, it reads everything. Embedded within the DNA are junk codes. These are sections of triplets that don't actually code for anything. They are just there. These are referred to as introns. And when the messenger RNA is removed or detaches itself, the polymerases are going to come in and remove those introns. So those introns are going to be X'd out, and the exons, or the reading code, is going to be spliced together. So you might start off with a chain of like 120 nucleotides, but end up with a chain of like 60 once all the junk's removed. There's some terminology involved with this. There's a 5' cap and a 3' poly A tail. Basically, the 5' cap is where we're going to start reading this RNA, and the 3' tail is what's going to be at the very end, and it's going to help prevent the messenger RNA from degrading, from breaking down, basically. Translation is going to be the reading of said messenger RNA and creation of the amino acid chains, and as I mentioned, requires all three, and that is going to take place within the cytoplasm, either within a free-floating ribosome or a ribosome embedded in rough ER. Transfer RNA can be charged or free. If it is charged, it means it has an amino acid attached to it. If it's free, it means it does not. That's all that's referring to. This is what transfer RNA looks like. This anticodon is basically going to be the opposite of our messenger RNA and is what's going to be attached to the messenger RNA, allowing for these amino acid that's attached up here to attach to the amino acid that's next in the chain. A ribosome is going to have two binding sites. There's a P site and an A site. The P site is going to hold the transfer RNA with the last amino acid that was added. The A site is going to hold the transfer RNA with the amino acid that is next in line. So, I believe I have a picture. I do not. This is about the point where I draw on the board. Destination of proteins. So the very, very beginning of the protein chain is what we refer to as a leader sequence. And that leader sequence will be red, and it will determine where it goes. If the leader sequence requires ribosomes, it will end up going to the rough ER and to the Golgi apparatus eventually. If there is no leader sequence, it will remain in the cytosol. So it will be used by the cell inside the cell. Reiterating that, in the Golgi apparatus, this is where the leader sequence is going to be removed because it does not code for anything. It's just, you need to go here, you need to go there. Any excess amino acids that aren't actually necessary are going to be removed, and if we need to add anything to the amino acid chain, do we need to add another protein chain? Do we need to add a fatty acid? Do we need to add a carbohydrate? Do we need to refold the chain? That's all going to take place post-translational, so after the chain has been formed, and it's going to take place in the Golgi apparatus. So packaging of proteins, it will first be in very small transport vesicles found typically in the smooth ER, and those transport vesicles will fuse with the Golgi apparatus on the trans side. It will then go through the Golgi apparatus, having all of its post-translational stuff happen to it, and eventually it will arrive on the cis side, will be packaged into other vesicles, and go to various places. Either it will fuse with the cell membrane and leave the cell, or it will go to another organelle to be utilized. RNA polymerase is what is going to encourage this messenger RNA reading. So in order to control protein synthesis, we actually want to control our RNA polymerase. RNA polymerase can be induced, so activated, or repressed, turned off. And this is going to be due to a variety of things that you just need to be aware are there. We're not going to actually touch on them. How do I break down proteins? I made too many. What do I do with them? We are going to use proteases. Proteases are enzymes that break down proteins, and we are going to have some specialized lysosomes called proteasomes that are going to contain these proteases. These proteases are not going to be free-floating in the cell, otherwise they would break down everything in the cell. So they're going to be contained within a pouch to protect the cell, and these proteases are going to remove these proteins, break them down. And these amino acids from the breakdown can then be reused. We have figured out that there's a special chemical tag called ubiquitin. Am I pronouncing it right? I have no idea. Does my pronunciation sound fun? Yes, so that's how I'm saying it. So ubiquitin is a chemical tag that will attach to proteins that need to be moved to a proteasome to be degraded. So this is how we know what ones need to be degraded. That does not mean if these proteasomes were to burst, these proteases would still attack and break down every single protein they came in contact with. And that is the end of chapter 2. Sorry about the 3 audio formatting. I just did not want to re-record my first 10 minutes. So this is 3 videos. It's approximately an hour and a half long all told.